All right. I'm going to go ahead and introduce our speaker today because it was me who wanted to invite her. So we have the privilege today to hear from Dr. Leah Starita at the University of Washington. Leah's awesome. We work with her in the Wagner lab. And the main goal of Leah's lab is to put an end to variants of uncertain significance, or VUS, to make genetic medicine more informative, equitable, and impactful. And the way they're doing this is by using multiplexed assays of variant effect, or MAVEs, to be able to inform variant classification to move VUS into more definitive classifications, such as, likely benign or likely pathogenic. We're working with the MAVE group at large to kind of pilot some oncogenicity classifications and try to use MAVEs to be able for in vitro and in vivo functional study evidence, especially within the new oncogenicity guidelines that are coming out. And I wanted Leah to come and kind of talk about the work with MAVEs and this process so that we could all get more involved. So Leah, thank you so much.
Thank you for inviting me. All right, so let's start the share screen process here. Okay. All right, are you guys seeing what looks like not presenter notes?
Yep. Yeah, looks
Okay,
great.
sounds good. And thank you, Wes, for inviting me. And this, I think it looks like a decent sized group here. So I would love it if, and I know that you guys are kind of doing somewhat similar stuff. And it would be awesome if you guys just wanted to interrupt and make it more of a conversation than a talk. So I think that would be great. So just feel free to unmute and ask questions as we go. Okay, so so we use multiplexed assays of variant effects, and I will talk about what those are. But first, I usually use this example to set up the problem. And again, this is in the cancer sphere, so it would be very similar to, very familiar to you all. But it's just that trying to frame the problem here. So this is a little cartoon of BRCA1, which is the tumor suppressor for hereditary breast and ovarian cancer. Um, and if we look at just these three little codons, what can happen as a result of a genetic test for BRCA1 is that you could have no change in the sequence at all, which is good. You can have a frame shift, or you can have a single nucleotide change that creates a nonsense codon. And obviously, those are fairly easy to classify as loss of function. However, if you end up with a single nucleotide change that changes an amino acid, those are really hard to predict. And they're even, still hard to predict. Even alpha missense overpredicts pathogenicity. So, what are these? These single nucleotide variants become these variants of uncertain significance. And this was the contents of ClinVar last January. I need to update my slides for 2024. But if you look at ClinVar, which holds all of the basically, genetic findings from genetic testing, you see that for missense variants, there's a small set of variants that have definitive classifications like pathogenic and likely pathogenic, and on the benign side as well, but about 80% of them are variants of uncertain significance. And this is actually a growing problem. So let me see if I can, it's blocking my slide here. This is actually a growing problem. So if you take the contents of ClinVar every January and you look at the number of VUS versus definitively classified variants, you see that it's growing. The number of VUS are kind of growing exponentially. And I think that what we think this is a representative of is that, as we sequence more people, we're just finding more rare variation. And so if you actually multiply the number of humans on Earth with the de novo mutation rate, something like 100 people are walking around with every possible single nucleotide variant that is compatible with life. And as we sequence more people, we're just finding those. And these rare variants are the ones that are, these rare missense variants particularly are becoming these variants of uncertain significance. And what are they, and they're rare. So a lot of this evidence that is typically used to classify genetic variation just doesn't exist, right? So patient phenotype can be used, but not for something like breast cancer, where there's a high rate of it in the population. Segregation analysis just doesn't really exist. Insurance companies don't pay for cascade testing. And then population-based data where, is really biased. So you can tell that you can classify a variant as benign if it occurs more often than the disease you're, testing for in the population. But since a lot of populations have been basically excluded from all of these big genetic studies, like that are aggregated in, say, NOMED or in TopMed or something like that, this data just really isn't useful for folks of Hispanic and African descent. So, the people who are, so VUS rates are actually higher in those populations, which, is an equity problem. And then, of course, you can use predictive models, so in silico predictions, and and that can be scaled to the entire genome, which is great. We're not going to talk too much about predictive models today, although I will say that they are getting better, and I think the combination of that plus functional assays, and again, functional assays can be used at almost any base in the genome as well, to scale to kind of the size of the problem. And when a functional assay is, I'm actually talking about, it could be anything from making a mouse model down to, in vitro assays where you might make the protein and test its, enzymatic function or something like that. And I'll be talking a lot more about those today and how we plan to scale these to kind of the size of the VUS problem. All of these types of evidence can be used at different weights. So strong, moderate, or supporting. I assume that I should know this more, but I assume that there's something similar for the oncogenicity evidence. But functional assays can also be used as strong evidence, which is important for actually, being able to move some of these variants to the more definitive categories. And then, again, these are just the 5 categories that you get. Okay, so historically, functional data can be generated for, almost any base change in the genome. However, as I just told you, there are, 500,000 variants of uncertain significance in, all sorts of genes that are known to be linked to disease, and that was a year ago. There are more than 1.2 million variants this year, right? So this problem is huge. All of these proteins, and these are missense variants, these are in protein coding genes. All of these protein coding genes kind of, they're all sorts of shapes and sizes and functions. And typically these experiments were done on, one postdoc or one graduate student would, functionally characterize a dozen of these variants. And this data would be this data would kind of come, 10, 20, 30 years after these variants were first seen in the clinic, right? And this is not going to keep up with the problem. But in the meantime, with the next generation sequencing and advances in the ability to write DNA, I've been part of a field that's been developing these multiplexed assays of variant effects. And this should say that so we can generate functional data for thousands of variants in a single experiment. And the idea behind this is that you take a library of genetic variants, and we try to make generally every possible single nucleotide change or every possible amino acid codon swap, and we get those into cells. These could be bacteria, these could be human cells. And then we apply a selection on the function of that protein or that coding gene. And again, these also work for non-coding variants as well. So this could be cell growth if your protein or regulatory element of interest is required for cell growth. We can use fluorescent reporter assays to separate cells that either have a lot of function or little function of a protein or regulatory element. And then from this data, we generate what we call a variant effect map, where we can take any single nucleotide change and understand its effect on function. And I'll go through a little bit more of what that means in a second. And I will walk through a specific MAVE for BRCA1 next. So BRCA1 is a tumor suppressor, but it's also essential for growth. So it's a little bit of a weird tumor suppressor. It's a double-strand break repair enzyme. It's in the double-strand break repair pathway. And we have this really great cell line called HAP1. It's a haploid cell line, which is great for genetic screening. So it's kind of the yeast of human cell lines, if you will. And BRCA1 is exquisitely essential in this haploid cell line. You can imagine a haploid cell line might be super dependent on its double-strand break repair pathway. And so what we're going to do is we're going to build an editing-based assay where we make variants in this haploid cell line in the germline in the genome of in the endogenous copy of BRCA1, and then we just wait to see which variants are becoming depleted from the population, and we use sequencing to track that. We'll go through that in a second. So the way this works, this is called saturation genome editing. And it works by you express, this is a model of BRCA1. It's got 23 exons. And we're trying to make every possible nucleotide change in exon 18 here. So what we do is we express Cas9, which causes a double-strand break in the exon. We also give these cells plasmid-based repair templates. So each repair template is a library, right? So we're transfecting in thousands of these. Each repair template has a different single nucleotide variant, either in the exon or right outside at the exon intron boundaries. And as the cell repairs this double-strand break, these single nucleotide with these plasmids, the single nucleotide variants become incorporated into the genome of the HAP1 cells. Again, it's haploid, so you only get one, so that's really handy there. And so then each cell will be expressing a different variant of BRCA1. And then again, we just wait, right? So because this gene is essential, we wait to see which of these variants are becoming depleted. And we track this by sequencing over the edited region. We sequence at day 5 to see which edits we actually made. And then we sequence again at day 11 to see which edits are becoming depleted from the population, or which edits are, just kind of growing nice and steadily. Each 120 bases or so requires a new guide and a new library, given the vagaries of homologous recombination and sequencing limitations, right? We try to sequence each base twice, so the length of Illumina sequencing is kind of limiting here. But in this project, we targeted about 4,000 possible single nucleotide variants in BRCA1. We were able to generate scores for almost all of them. So the scores from this assay are around 0 with no log 2 of 0 with no change. You get your synonymous variants, which makes sense, right? There's no change, but about 1% of those actually cause a big enough splice defect where they kind of end up down here with the nonsense codons. So a log 2 fold change of -2 is where like the nonsense codons hang out, those canonical splice variants on both sides of each exon, although some of those seem to be perfectly functional, which I know a lot of people don't like to hear. And then the missense variants are bimodally distributed across these two distributions, which is what we kind of expect to see, with about 20% of the variants behaving kind of nonsense-like, so loss of function. And we classify variants that way. So either they behave like the synonymous variants or they behave like the nonsense variants. And again, I just told you that we use this crazy haploid cell line. It used to be leukemia. What does this have to do with hereditary breast and ovarian cancer? Are we measuring the same thing? And so we took all the variants that were already in ClinVar, which are about 200 of them. And again, BRCA1 is cool like that because it has all of these known pathogenic and benign variants. And this becomes a problem a little bit later that we can discuss. And this assay separates those variants really well. So all the likely benign, most of the likely benign and benign variants are in the functional category or score in the functional category, whereas the likely pathogenic and pathogenic variants mostly scored in the non-functional or loss of function category. And so with this assay, it's pretty accurate for separating variants of known function, which is with known functional effects. And the ones that are wrong, everybody asks me about this. Some of these, so as you guys know, ClinVar takes all comers. Some of these were actually misannotated in ClinVar. Some of them are actually, one or two of them are in a splice, are in those canonical splice positions, but they're actually upstream of the start codon. And it turns out that at least in this cell line and potentially in humans, that those canonical splice variants are perfectly fine. But given the rules of classifying variants, they get, a kind of an automatic pathogenic. So I think those kind of need to be looked at further in patients. Okay. So again, we create this variant effect map for all of BRCA1. So all the data I've shown you up to now has been aggregated, but here you get this really nice kind of map of the function of BRCA1. So this is exon 2. Every column is a position in the exon. The coloring is the type of variant. So these green ones are the missense variants. These yellow ones are the canonical splice variants, and you can see these are the ones that are have no change in function upstream of this is the start codon, ATG. And if these little blocks are colored in, it means that it was a loss of function variant. And so we did this for all 13 exons that we targeted in BRCA1. And we can use this map to kind of look up functional effects for a variant that might be seen in the clinic. So for example, you can look up this mutation, methionine 18 to lysine, and you would say, hey, that's filled in, it's functionally abnormal. Whereas, if you happen to see leucine 1843 to isoleucine in the clinic, you can kind of just look this up and it would be functionally normal. So this is what we, kind of hope to build for a lot more proteins that are, or coding genes that are linked to disease. Okay, so I told you the point of doing this is to reclassify variants of uncertain significance, and you can't really just use this raw data in a variant classification. So how do we do that? How do we translate kind of raw data into functional evidence that can be used in the clinic? And the way we do this is using, first of all, this project was led by Sean Fair, who I think Wes and Jeremy might know. And he's a genetic counselor. And he was, working in the clinic and was actually really tired of returning variants of uncertain significance to his patients. So he came back to graduate school and he's a PhD candidate in jointly advised by myself and Doug Fowler, and he led this project. So we had all of this functional data for BRCA1. We also had functional data that was published previously for P53 from Dana-Farber and P10 from our group and another group in Oregon. And the first thing he needed to do was, figure out what would be the strength of evidence that could be used. How much weight could this functional data carry in a clinical variant interpretation framework? And ClinGen had just come out with their guidance for using functional data. And what that guidance said was that if you take all of these variants of known effect from ClinVar, depending on how many of them it gets right, you can use a Bayesian framework to figure out whether or not it should carry strong, moderate, or supporting evidence. And this data, because there are so many variants of known effect in ClinVar for BRCA1, and because the assay separated them so accurately, we were able to give the BRCA1 data strong evidence. It's a calculation called the odds of pathogenicity. And so what we did was we then did a kind of a reclassification exercise. We collaborated with Ambry Genetics. They were wonderful because all of the other data, all of the clinical data that you need to do a reclassification exercise doesn't exist publicly. So that's why we had to collaborate with the company. They were wonderful. They gave us all the data that they had for all of the variants that overlapped our data set. And you can see that there was about a third, or a little bit more than a third VUS. And so what we did was we took all of that data that they gave us, all of the functional data that from the Finley paper. We added strong evidence for the loss of function variance toward pathogenicity, strong evidence for the functional variance toward benign, and we just used like the vanilla ACMG framework, which is actually not how most of these genetic testing companies work. But even just doing that, we were able to reclassify many of the variants. So about 50% reduction in total. A few of them moved from VUS to likely pathogenic. And as we expected, actually, many of them moved from variant of uncertain significance to likely benign. And Ambry Genetics then went and recontacted all of the providers who had gotten VUS results in the past and updated their results, which is really exciting to us because we had kind of to Doug and myself, because we had been involved in actually developing these assays, and kind of we've gone all the way from developing the technology to it affecting bench to bedside, if you will, which is really exciting. So again, we had published data for P53. We were able to reclassify about 70% of the VUS in that gene, which is exciting. Kind of more of those went to likely pathogenic, which I thought was kind of an interesting result. P10, only about 15% of the VUS were reclassified. And P10 is kind of an example of a problem I was telling you before. So for this exercise to work, you need a lot of control variants, you need a lot of benign variants, and a lot of pathogenic variants that kind of already exist to kind of do this calibration to understand the weight at which you can use this data in this variant interpretation framework. There's like 2 known benign variants in P10. This gene just does not have a lot of benign variation. And so you can't really understand the specificity of the assay. And so therefore you kind of have to, we use the VSEP rules here, and we have to cap the strength at which we can use the P10 data, and we really can't use it as evidence toward benignity at all. Like if the protein's broken, we can use it as evidence toward pathogenicity. So, that's kind of one of the limitations for sure. And we're working with ClinGen to kind of see if we can figure out other ways to validate some of these assays, to understand, to be able to use them, because it turns out that in ClinVar, 70% of the labels are on 7 genes. So when you get away from BRCA1 to P53 and a couple of more genes, you're just not going to have the variants of known effect to be able to do this exercise. But it's not and then it's not just our lab who is doing this. So there's a great MSH2 data set. MSH2 is one of the genes that is mutated in Lynch syndrome. And there's a really nice data set out of University of Michigan, the Kitzman lab, and they were able to reclassify 75% of the variants, again, most of them going from VUS to likely benign. And then there was another study also using saturation genome editing out of the Sanger on DDX3X, which is a gene known to be involved in intellectual disability and developmental delay. And 90% of the VUS were reclassified in that study. So this is working, right? We're able to take, even where it kind of doesn't work, which is in P10, we're able to, at least move some of the variants. And in some cases, we're able to move a really large percent, right? So 50 to about 90% in some of these other genes. And so this is, this is working. So what's the goal? So how do we take these multiplex assays of variant effects and these variant effect maps and bring them to clinical use, right? And okay, so what's the vision? How do we get there? How do we get from having this technology to no more variants of uncertain significance? And the first is scale up, right? So we have some of these assays that are highly accurate, like the saturation genome editing that I just showed you. We have some other assays that are pretty specific for identifying loss of function variants. And I'll explain what that one is in a second. That can be applied to a lot of different proteins. And so we kind of have, we have the machine, we just need to kind of turn the crank. And that's the first thing I'll talk about. And we're doing that. We're doing it personally through the IGVF, the Impact of Genetic Variation on Function Consortium from the NHGRI. So Doug and I have a production center, a data production center. And we're performing saturation genome editing, which is that assay for BRCA1 that I just showed you, across a lot of different genes that are essential in the cell line and also linked to disease. And we're also performing another multiplexed assay of variant effect called VAMP-seq, which measures protein abundance. And that is a really simple assay where you take your gene of interest and you fuse it to GFP. And variants that are destabilizing cause the GFP to become unstable. And so by FACS, you can separate out variants that are stable versus variants that are destabilized. And a lot of pathogenic variants are pathogenic due to kind of unfolding the protein. And this assay has a really good positive predictive value, not so great on a negative predictive value, but hey, we'll take half of that. And so in the beginning of the consortium, we spent a lot of time kind of optimizing these assays for production scale and making them more robust. So here we've got just a little bit of snippet of data for PALB2. We're getting really good at it for saturation genome editing. And again, just like some new engineering new landing pads and stuff for the VAMP-seq assay to be able to get genes into the cell line kind of more efficiently. We adapted a lot of our kind of upstream reagent creation work to automation. So this is 96 guide transformations, for example, 96 different guides in one plate, which is super fun. This little robot called a Mantis that can put your enzyme solutions in 384 well plates. It's pretty awesome. And then we choose genes for these assays for different reasons. So for example, some of these are PALB2 is the third gene in line after BRCA1 and 2 for hereditary breast and ovarian cancer. And we choose a lot of these by kind of their clinical importance, I guess. So many of these that are labeled ClinGen are on the ACMG 78 list. So these are the genes that have to be, if a pathogenic variant is found in them, incidentally, through exome sequencing or some other type of sequencing, they need to be returned to the individual because medical intervention can actually ameliorate the risk of disease. And so therefore they're sequenced a lot, and there's a lot of variants of uncertain significance in all of them. These ones labeled Gregor. So I work with the Gregor Consortium, which is the NHGRI's a rare disease consortium. And so we perform functional assays for them when they have, a gene of interest like CTCF, which is just a really interesting gene, where they're finding de novo variants, for example, in autism cohorts. And then some of these other genes are potential Mendelian disease genes. And we're kind of doing these assays or performing these assays in support of new gene discovery, and also phenotypic expansions for these genes. We also have this assay working in primary T cells, the saturation genome editing assay, so we can look at genes that are important for immune phenotypes. And then also the protein abundance assay. CPIC is the Pharmacogenomics Consortium, so it could be really useful to know which genes affect drug metabolism, for the gene products that affect drug metabolism, which variants are going to be loss of function or not, to help figure out, which people might be more or less sensitive to some of these drugs. So CYP2C19 and G6PD kind of fall into that category. Hereditary breast and ovarian cancer, again, is actually just my my own interest. But anyway, so a lot of these genes, we perform a lot of these genes kind of in service to different consortia for the NHGRI. Okay, so we have assays that work now, but we also need new technologies. So I was telling you about that list of ACMG genes. There are many genes that kind of aren't expressed in some of these kind of cancer cell lines we tend to work in because they're easy to work on. They happen to be haploid, or they just grow really well. And an example of that gene I'm going to talk to you about in a second, but there are many genes that are important for disease that are only expressed in very kind of terminally differentiated cell types that we don't have access to at the moment. So the idea here is, can we adapt these multiplex assays of variant effects to induce pluripotent stem cells so that we could, maybe make a variant library in the stem cell state, differentiate to, the cell type of interest. In this case, we're just kind of showing a cartoon of cardiomyocytes, erythrocytes, or neurons, and then phenotype those variants, right? Does it have an effect on cell survival? Does it have an effect that we can measure by flow cytometry or FACS? Or, for example, you can imagine doing an assay where single cell RNA-seq is the readout. And again, this was developed by Sean Fayer, who is a quite prolific graduate student. So in this case, we're using MYBPC3 as a test case here. So MYBPC3 variants cause pathogenic variants can cause hypertrophic cardiomyopathy. Pathogenic variants in this gene are about 50%, are responsible for about 50% of inherited HCM. Hypertrophic cardiomyopathy is just a thickening of the walls of the heart, which can cause heart failure. This gene is kind of this yellow snake here, which is part of the sarcomere. It kind of connects the actin to the myosin. Again, myosin binding protein C. And we're going to be talking about this C10 domain here. So the C10 domain is required, function of the C10 domain is required to get this big protein into the sarcomere and functioning as kind of a brake. And again, unfortunately, most human cell lines are annoyingly diploid. So these IPS lines are diploid. So how do we deal with that, right? So MYBPC3 variants are pathogenic variants. It behaves in an autosomal dominant fashion, right? So we want to get a wild type version. So what we do is we knock a wild type construct into the cell line that is fused to GFP. And then in that line, we delete a little piece of an intron that no longer has a guide site in it, so that we can target the second allele only with a couple. This is at the 3' end of the gene. We come in with exons, I think 32, 31, exons 28 down, and then that is fused to a red fluorescent protein, and every possible variant is in this case, exon 32, so about 500 variants. And another problem with IPS lines is that prior to, a whole bunch of work by companies like, Stem Cell, oh God, and Synthigo and things like that, using Cas9, creating double-strand breaks in these lines was like highly toxic, but there's been a lot of work, not by us. We've been able to kind of take advantage of it, where we're seeing using a lot of different products that you can buy from stem cell technologies, for example. You can reduce the toxicity of Cas9 or of double-strand breaks. So that's been great. And in the literature, the biggest libraries that we had seen in pluripotent stem cells, there's about a dozen variants. So we're really worried, right? So this library is about 500 variants. How many pluripotent stem cells were we going to get? And then because of this toxicity, how was were we going to be able to differentiate these pluripotent cells into cardiomyocytes, which, if you know about this, it's actually one of the more difficult differentiation protocols. Okay, so is this working? So again, we tried to edit 500 variants into these cells. We have edited iPSCs. We differentiate into cardias. You can see they become kind of big and boxy. The GFP allele is attached to the wild type copy of MYBPC3, and you can see that you get this nice striation of the sarcomeres. And then the apple, or the red floor, is tagged to the variants, and you can see one where you get really nice co-localization with the wild type, and then others where you get like kind of nothing. And again, this, we don't know which variant this is, but this was a good indicator that we had variants that were having differential effects, which was exciting. And again, MYBPC3, this region that we're targeting, is important for, for MYBPC3 to be incorporated into the sarcomere. When it's not incorporated into the sarcomere, it gets degraded. And so we're able to sort on this phenotype, this fluorescent phenotype. So variants that cause, no defect will be in the high bin, and we sequence the variants in that bin. And then variants that cause, destabilization will be in a low fluorescence bin, and we sequence the variants in those bins. And then we just create a score by basically creating a weighted average to try to, by where the variants are across these fluorescent bins. So a high scoring variant will be stable and expressed, whereas a low scoring variant will be unstable and not expressed. And so, first of all, we were super excited because we got back out of these cells about 493 of the 499 possible variants. So we were super excited about that. That was a big worry going into this. We had done a lot of work on this engineering to do this, and we were able to get the scale that we needed. And then we were also able to stratify these variants by function, which is pretty exciting. So the synonymous variants are high scoring, generally, or higher scoring than the nonsense variants that are down here in the canonical splice variants. And these missense variants kind of span this functional effect, right? So some of them behave like nonsense variants, and then some of them overlap the synonymous variants, which is pretty exciting. And then again, importantly, this is a cell line that we have, an IPS line that we have kind of badly abused at this point. How are the known pathogenic and benign variants behaving in this assay? And we're able to separate those pretty well, where the pathogenic variants are lowly scoring and the known benign variants score higher than that. So we're able to separate these variants. And that's really exciting. So we are taking this forward to generate data for the entire C10 domain of MYBPC3, and then also apply this to other genes that are known to act in specific cell types. So for example, POLG in neurons and RPS19 in erythrocytes. So this is really exciting, and we're hopeful, we're kind of building on this technology now. Okay, so we have scale, we have new technologies. The next thing we need to do is kind of build the community. And this is something I know is like near and dear to y'all's hearts. How do we build, minimal information standards, reporting these assays, things like that, which make these sorts of data kind of more usable and reusable and extensible to more people. And so the way we've done that is Doug Fowler and I, along with some other, friends of your lab, so like Alan Rubin, and then some folks from the Sanger, founded the Atlas of Variance Effects Alliance. And this is some of our propaganda here. So you kind of see these people who are kind of walking through maps of protein function. This is like would be the dream here, where you, for each gene of interest or each region of interest, again, non-coding is very important. You would have a map that existed prior to a variant having that variant identified in the clinic. Right. And so we founded this in 2020. We've got 500 members from 105 institutions in 30 countries. We support the Mutational Scanning Symposium that I know Wes was at last year. This is a great meeting where we try to bring together people who are going to use the data. So the clinical folks, variant curation experts, and also the people who are generating the data so they can discuss together. We support the Variant Effects Seminar Series, which I believe there is a talk happening in 15 minutes. This is a seminar series for trainees that is also organized by trainees, which is great. And we have 4 interconnected work streams. And what those work streams are doing, again, is trying to build, they just published, as a preprint, we've got some minimal information standards for how to publish these assays. I'm the co-chair of the Clinical Variant Interpretation Group, where we try to develop guidelines for using this assay or using these data in variant classification. It's exciting. We also build tools for the community. So importantly, I work with Doug Fowler and Alan Rubin to host MAVEDB. So this is a database that holds all this data. Right now, it was really built to support like kind of data science uses. So for example, if you were going to try to develop the next machine learning algorithm to predict functional data effects, you could, the effects of mutations, you would download entire data sets, for example. But going forward, the people who have been yelling the loudest to fix this database have been the clinicians. So what you can't do with this database right now is search for your variant in particular. And the clinical community does not like that. So the next thing we're trying to do is really clinical outreach. Again, clinical users can't really use MAVEDB. They don't like it when they ask where the data is, and I say supplementary table 2. Clinicians don't have much time, right? They need to be able to look for a variant, and they need to be able to make a quick decision. ClinVar is the place where many clinicians will go look for data in general for a variant. ClinVar will hold functional data, but right now it's kind of a mess, and it's actually a spectacular mess. This is some of that BRCA1 data that I actually uploaded. It's all, you can see it, the formatting's all jacked up. You're allowed one unstructured sentence right now. So if I submit data and somebody else submits data, it's going to look completely different. So we're working with ClinVar right now to build a better data model for MAVEDB or for holding functional data. And we're also working with them to connect MAVEDB, to federate MAVEDB with ClinVar to be able to feed data into ClinVar and also pull ClinVar data back out in order to make visualizations for clinicians that will, kind of make sense, kind of be able to give them a dashboard for functional assay results. It's exciting. And we're trying to learn from clinicians. So if anybody out here in this audience is using MAVE data, we want to hear from you. So we've got a really great, UW Medicine has one of the oldest and largest genomic medicine departments. And we're trying to learn from them kind of how they're using the data. And if our neighbors aren't using the data, we know we've got a problem if we can't get them to drink the Kool-Aid. So, we're really working together with that group. And then we're also trying to learn from them to build a curriculum, like a training curriculum, like Wes was saying, where they hold hackathons and things at different conferences. We're hoping to be able to hold training sessions for people who are making these clinical decisions on how to use functional data. Let them know that it exists, let them know that it's starting to exist at a scale that it's going to be useful to them, and then, how they can take that information and apply it to variant classification, known to variant classification frameworks. So, we've got scale, we've got new technologies, we're kind of doing a lot of community organization and clinical outreach, and that's kind of our plan to get from these technologies, right? And that's where we kind of started at the bench all the way to hopefully making a big impact in genomic medicine. So with that, I've got a lot of people to thank. My lab, I work in a lot of different consortiums. I work very closely with Doug Fowler and other PIs in our IGVF center, Alan Rubin, and I've got a huge team. So we've got a software development team, which is really exciting to be working on MAVEDB. And lots of folks in the lab who are, generating tons of data and new technologies. So with that, I will happily take questions.
Leah, that was a great talk. It looked like most of the work that you've been doing has been with hereditary genes, hereditary breast. Are there plans to look specifically at somatic? Obviously, like BRCA1, BRCA2, PALB2, et cetera, are implicated in somatic cancer as well, but I'm wondering.
Yeah, so obviously some of the tumor suppressors play a dual role there.
Of course, yeah.
Yeah, so it's a project I didn't talk about. We actually have a really great, we've been developing a prime editing assay to look at drug resistance for oncogenes. And that works pretty well. There are other groups out there. I know the Broad has a ton of data sets on kinases that they haven't released yet. I don't know what they're sitting on them for. So to throw them under the bus. Yeah, there are a lot of groups out there who are really interested in drug resistance and also oncogenes. It's just not, I'm a hereditary breast and ovarian cancer girl myself.
Yeah.
So it just depends on the lab's interest. And, that's one of our community organizing things is we would really like to reduce the activation energy for people to do these assays. Right. So can we develop ways where they'll be able to disseminate their data better? Can we try to have like kind of a no code analysis pipeline sort of thing? I think people, so that we can take people who have the domain knowledge of genes like you're talking about, and then start, kind of multiplexing, go forth and multiplex, as George Church would say.
I was also going to ask, sorry, one other question. Yeah. What kind of work you're doing with groups like AMP and ACMG to incorporate into the guidelines how these should be used and with what strength? And
yeah, we work more with ClinGen's sequence variant interpretation group.
Okay.
And I feel like they're kind of it's kind of a roundabout where they're kind of more plugged in with ACMG and AMP. We actually have a survey right now that's about to go out to AMP, and those groups to try to understand how people are if they're using functional data and how and what their barriers are.
Yeah. Yeah, because I think it's so important and so relevant and really important for the community to kind of come to agreement, because I think there's a lot of disagreement on how much weight to put into high throughput assays.
Exactly. And again, that's one of our biggest problems, and again, BRCA, I wrote my thesis on that almost 20 years ago, but, and it's a favorite gene of mine, but one of the reasons we started there is because that's where the control variants are.
Yeah.
And so kind of as we move into, genes where those variants just don't exist. PALB2, for example, it has no classified missense variants, except it's got some benigns, but that's it. So like I've now generated this 13,000 variant data set. What the heck am I going to do with it? So it's a question near and dear to my heart.
Yeah, Cheryl, I would recommend, if you have genes of interest that you want done, you can go. I dropped a link in the chat for the MAVE registry, so you can go and you can actually make nominations.
Okay.
So like I kind of mentioned at the beginning, we curated a list of like 20 genes that are both gain of function and potential loss of function, where we kind of, as a clinical team, have some struggles in trying to actually classify these for our rare pediatric cancers. And so we dropped those nominations in there. And then, we'll be working with, the MAVE Alliance to try to actually figure out how to use this at NCH for functional evidence. And something that's really, near and dear to my heart is trying to you have, things like hackathons where we actually talk about, how could we use these as functional evidence, and trying to like pilot some of those studies with some of these genes. And I would definitely love to, have more people kind of contribute and work on that together.
Yeah, MAVE registry, I should have talked about that with this group. I'm sorry. I just gave a talk to biochemists and they were like, what? So I took it out. So MAVE registry is another AVE kind of tool. One of the ways we use it is to, kind of direct traffic for, who's doing what gene. And the other way is nomination. So if there are genes, just like Wes said, that, you would love to see some functional data for, just nominate it. And we're trying to build potentially like a matchmaking service as well, like matchmaker exchange for rare disease, but this would kind of be a little bit different than that.
Thank you. Great talk.
Okay.
Anyone else have any questions or comments for Leah? All right. Well, Leah, thank you so much for the wonderful presentation. Glad to have you here.
Great. Thank you for the conversation this morning. All right.
All right. Thanks, everybody.